Hello everybody, and welcome to the Meet the Genes in Space Toolkit webinar series. I'm Emily Gleason from Mini PCR Bio, and today I'm going to be talking to you about the P51 fluorescence viewer. Just to give you a quick summary of today's webinar, we're going to start out with an overview of the Genes in Space contest. Then we're going to introduce you to the P51 fluorescence viewer, the subject of today's webinar. And finally, we're going to hear from 2020 Genes in Space winner Christoph Mosquitta, who is going to be telling you about how he is going to be using the P51 fluorescence viewer to perform his Genes in Space experiment on the International Space Station later this year. So to get started, what is Genes in Space? Genes in Space is a science contest that invites students in grades 7 through 12 to design DNA experiments that address challenges or opportunities in space exploration. And the idea is that while NASA and other private companies are building rockets that may take humans back to the moon someday, or even to Mars, there's a lot that we don't know about the effects of these long-duration missions on the human body and on the bodies of the other organisms that we might bring with us. So Genes in Space is your chance to ask questions that you might have about how spaceflight might impact biological systems, and then to develop an experiment that will help us address that question and potentially help us keep astronauts safe or perform other functions on a future long-duration space mission. And I've put a few examples of questions that you might ask up here, anything from how spaceflight might affect humans' DNA to how microorganisms might react to life in microgravity. But what we want to know for Genes in Space is what's the most relevant, creative, and bold question that you can ask. And this isn't just a thought experiment. The grand prize of the Genes in Space contest is that your experiment will be performed on the International Space Station. So if you are selected as the winner of this year's Genes in Space contest, we will work with you to adapt your experiment and perform it on the International Space Station sometimes next year. And this will allow you potentially, if you're a winner, to have an impact on our understanding of biology in space. Just to give you an example, Finsum Samson and Yuji Wang applied to the 2019 Genes in Space contest. They were interested in how microgravity and other space conditions affect astronauts' nervous systems, and they won the contest that year, and their experiment was performed not even 2 months ago by astronaut and virologist Dr. Kate Rubins aboard the International Space Station. Then their samples were shipped back to us here on Earth for further analysis, and we can confirm that their experiment was a success. And applying to Genes in Space is easy. The application form is now open. It's an electronic form that you can use to submit your proposal. It can be found at www.genesinspace.org/apply. And you must submit your application no later than the end of the day on April 12th of this year in order to have it be considered for the 2021 contest. And applying to Genes in Space is free. You don't have to have any equipment or perform any experiments. You simply have to have an idea for a DNA experiment in space and write up a short proposal telling us about that idea. So the first part of the proposal is to identify an open question. What is something that you're wondering about in terms of how life is impacted by space conditions? And then you want to do some research and formulate a hypothesis, what's known about your topic here on Earth and potentially in space, and use that information to formulate a hypothesis. The next thing you need to do is develop an experimental plan to address your hypothesis that uses the Genes in Space Toolkit. And finally, you want to justify your use of the International Space Station. So what is the Genes in Space Toolkit? This year, it consists of 3 tools: PCR, the BioBits cell-free system, and the P51 fluorescence viewer. My colleague Allison told us all about PCR in an earlier webinar that I've linked in the description below. So if you're interested in that technique, you can click on the link below to check out her webinar. Similarly, my colleague Allie told us all about the BioBits cell-free system in another webinar that I again have linked in the description below. So I encourage you to check out that webinar as well. And finally, the P51 fluorescence viewer, which is the subject of today's webinar. So to tell you a little bit more about the P51 fluorescence viewer, we're first going to talk about what is fluorescence, next, how fluorescence is used, and finally, introduce you to the P51 fluorescence viewer itself. So what is fluorescence? When we look at the world around us, we perceive objects as having color. And the reason for this is that visible light contains all of the colors of the rainbow. And when it hits an object, most of those wavelengths of light are absorbed by the object, but some of them may be reflected. And the light that gets reflected is what we perceive as the color of the object. So to give you an example, when I look at this apple, visible light is hitting the apple and most of it's being absorbed, but light in the red portion of the spectrum is being reflected. So I see that reflected light and perceive the apple to be red. Fluorescence works a little differently. Fluorescent molecules absorb energy from certain wavelengths of light and then emit that energy back out as longer wavelengths of light. So instead of just reflecting light, they're actually emitting longer wavelengths of light. And that's why fluorescent molecules appear to glow. And fluorescence can be found everywhere. Many substances in your house, many common household substances may be fluorescent, and different biological systems produce fluorescent molecules as well. One of the most commonly known examples is that of the green fluorescent protein produced by certain kinds of jellyfish. And it's thought that these jellyfish use this fluorescent protein in order to attract prey, but scientists have actually taken this protein out of jellyfish and have used it for a number of applications in biological science to better understand biological systems. And that brings us to our next point, which is that fluorescence is a useful tool for allowing us to see biology. So DNA and protein and other molecules that make up biological systems are really, really critical to the function of those systems, but they're typically not visible to the human eye, and which makes it really, really hard to study these molecules. However, we've come up with various techniques in order to make these molecules fluoresce. And when we turn these molecules fluorescent, we're able to see them and therefore able to understand how they're functioning within the cell. So fluorescence helps us by taking molecules that are not normally visible and making them visible and therefore easier to study. And I'm going to give you a few examples of how we use fluorescence in biology. These are examples that most commonly are used here on Earth, but they may be applicable to space as well and potentially something that you might want to use in your Genes in Space proposal. So the first example is biosensors. So biosensors are devices that use a biological component to detect the presence of a particular molecule. And that molecule might be a contaminant in a water sample, for example. It might be a specific type of protein, or it could even be a specific DNA sequence that you're interested in. And when that molecule, whatever it is that you're interested in, is present, the biosensor produces a signal that can be visualized or otherwise measured. So in the case of fluorescent biosensors, the biosensor would produce a fluorescent signal that you can detect. And here I'm depicting a really simple biosensor that takes place in a test tube, but you should know that some biosensors are included in machines or may even be affixed to clothing in some way. So when the molecule that we're interested in detecting is added to my biosensor, a reaction occurs that results in the production of a fluorescent signal. So when that molecule is present, our test tube biosensor begins to glow, and that lets us know that the molecule is there. So we couldn't see the molecule, but the fluorescent signal tells us that it's present. To give you a more specific and relevant example, biosensors are being used to detect the virus that causes COVID-19. So a number of biosensors have been developed that recognize specific sequences in the virus that are unique to the virus that causes COVID-19. And when those sequences are present, a fluorescent signal is produced. So you can add a sample to a tube, a biosensor in a tube, and then look to see whether or not that biosensor fluoresces or not, and that would tell you whether or not that virus is present in your sample. To give a more complex example, we're going to talk a little bit about qPCR, or quantitative PCR. So my colleague Allison told you a few weeks ago about PCR, which is another one of the tools in the Genes in Space Toolkit. And essentially what PCR is a technique that allows us to take a complex sample of DNA, find a specific sequence that we're interested in, our target sequence, and make billions and billions of copies of that sequence in order to be able to analyze it. As I told you previously, that DNA that we're making with PCR, that's not visible to us. So we have to follow PCR with additional laboratory techniques that allow us to visualize or otherwise analyze that PCR product that we've made. And one example here I'm giving you is gel electrophoresis. The point is that many of these techniques usually tell us whether or not a DNA sequence is present, but it doesn't tell. But what if you want to know more? So for example, what if you want to know how much of that DNA was present in your sample to begin with? Following PCR reaction with, say, gel electrophoresis doesn't answer that question. And I'm going to give you just a really simple example of why you might want to know that or why that information might be useful. We're going to focus again on viruses because they're particularly relevant to today's world. So in that COVID-19 testing example that I gave you previously, the question that we were asking was, is the virus that causes COVID-19 present in a sample or not? That's a yes or no answer. But in the case of viruses that are better studied, knowing how much of a virus is present in a sample can tell you something about the severity of the illness or potentially how contagious a person might be. One example would be the HIV virus. That virus has been causes AIDS and has been particularly well studied in recent years. And what scientists know is that if the level of the amount of HIV virus rises above a certain threshold, it tells you that the patient may be experiencing a more severe form of the illness, and it may be time to, say, change the treatment that the regimen that the patient is on, or take as other action to help the patient. And PCR, followed by a technique like gel electrophoresis, can't tell you how much HIV was present in the sample or not. It just tells you whether or not that's there. However, if we follow the PCR reaction as it's occurring, we can actually estimate the amount of DNA in our sample. And I'll tell you a little bit about how we're going to do this. So qPCR, or quantitative PCR, uses a fluorescent dye, but that dye only fluoresces when it's bound to DNA. And every PCR cycle, more and more DNA is made, and the fluorescence gets brighter and brighter. So as the PCR reaction progresses, more and more DNA is made, more and more of our fluorescent dye begins to fluoresce, and eventually it reaches the level at which we're able to detect that fluorescence. And if you start with more of your target DNA sequence, if more of your target DNA sequence is present at the start of your reaction, then the fluorescence will get brighter faster and be able to be detected sooner than if you start with less of your target DNA sequence. So if you follow the PCR reaction and look at when you're able to detect the fluorescence, that tells you something about how much of your target DNA was present in your initial reaction. The earlier on in the PCR reaction that you're able to detect the fluorescence, the more DNA that was present there to begin with. And that is a super useful technique, both for determining viral load in, say, cases of HIV, like I told you about previously, as well as measuring the amount of other DNA sequences for a number of different experiments. So those are just two examples of fluorescent techniques that we use here on Earth. There are many, many others that you might use in a Genes in Space proposal. We're not going to go into them here today, but fluorescence is, I hope I've convinced you, a hugely useful technique for visualizing things in biological experiments. However, the machinery that we use to detect fluorescence can often be expensive, large, and complex to use. Sometimes it requires specific training. And all of those things aren't very good if you think about a long duration mission to the moon or to Mars. Large, heavy objects are very expensive to fly into space, and it's hard to find room for them in the small capsules that we send out into the solar system. And additionally, if the equipment's really complex and requires special training, that's difficult for the small crew that may be going on one of those missions to receive that training. But if you think about it, fluorescence can be really simple. All you need is a light source that emits the type of light that's absorbed by the fluorescent molecule, and then a way to detect the wavelength of light that's emitted by the fluorescent molecule. And that's where the P51 comes in. It's a simple device that allows us to detect fluorescence in a tube. And it's very easy to use. I'm going to show you my P51 now. So it's small, it fits in the size of my hand, and it's pretty simple. It just has a switch on the back that turns on, in this case, a blue light. This blue light can be absorbed by many fluorescent molecules. And then when we close it up and we look in the front, there's a filter that filters out all the wavelengths of light except for the wavelength that's being emitted by our fluorescent molecule. So if you look closely at my tubes, you can see that they're fluorescing green. So very simple device, easy to use, and ideal for a space condition because it's small, lightweight, and simple. So that brings us to Christophe Mosquitta. He is currently a senior at Stuyvesant High School, and he's the 2020 Genes in Space winner. Fun fact about Christophe is he's a repeat Genes in Space applicant. So he applied to Genes in Space several times, the first of which was back in 2018 when he was a freshman. And back in 2018, his proposal was actually selected as an honorable mention in that year's contest. And here's a picture of Christophe a few years ago with his Genes in Space medal. And then Christophe went on to apply again in 2019 and again in 2020 when he was able to realize his dream of becoming the Genes in Space winner. And Christophe's winning Genes in Space experiment focuses on pharmaceutical drugs and how they're metabolized by astronauts in space. And what he found out was that actually several drugs have been shown to be less effective in microgravity than they are here on Earth, and nobody really understood why. And that's a really concerning problem because we're relying on pharmaceutical drugs to help keep our astronauts healthy while they're in space. So Christophe developed an experiment to start to address the question of why certain pharmaceutical drugs may be less effective in microgravity. And Christophe's going to join us here today to tell you all about it. So without further ado, I'd like to introduce you to Christophe.
And hey everyone, I can't wait to tell you all more about this awesome addition to the Genes in Space Toolkit. And I'm especially excited because just last year, when I was writing my proposal, I could only have dreamed of having such a quick and easy way to evaluate gene activity on the ISS. So specifically, I was trying to investigate genes in the liver, since that could help explain why medications tend to be less effective in space than on Earth, and also help us rethink how we treat illnesses in astronauts on long-term missions. The P51 is now at the core of my experiment, launching to the International Space Station later this year, and I think it could make a fantastic addition to your proposals too. But before I dive into how I used it, let's start with a little context. An important determinant of a medicine's effectiveness is how quickly the body can break it down. And so you can imagine that the faster this happens, the weaker the medicine's effects tend to be. Now crucially, this breakdown is carried out by specialized proteins in the liver. Medicine will attach, and it'll leave in some metabolized form. But here's the critical part. If the amount of this liver protein increases, then medicines tend to be broken down faster, and we already know that that tends to make them less effective. So clearly, the concentration of this protein is important, and it isn't unreasonable to think that it could be one of the reasons why medicines don't seem to be working that well in space. Now that's a great foundation for a proposal, and all I need to do now is to find a way to determine if the protein is being produced in an abnormal amount aboard the ISS. So how can I do that? Well, RNA is the intermediary between DNA, which encodes instructions for making proteins, and the proteins themselves. So if I can measure the amount of RNA corresponding to this class of proteins in space, that's a way of telling me how much of the protein is being produced. And believe it or not, that's it. That's how I can determine whether astronauts could be metabolizing medicines faster on the cellular level. And that potentially explains the reduced efficacy of the drugs that's been observed. Fantastic. Now, I can put this all together. So let's say I have 8 groups of cells, and I'm going to administer medicine in a different concentration to each of them. And in response, they'll all produce a certain amount of RNA. Now here I'll also throw in 2 controls, one where I add water instead of medication, and another where I add no treatment at all. So next, I'm going to isolate RNA from the cells, reverse transcribe it into cDNA, and design a PCR reaction for some gene of interest. Let's call it gene X. And this is going to be the gene that encodes the drug metabolizing proteins we talked about earlier. And while we're here, let's not forget that we also have to add DNA binding dye. That's going to help fluoresce in the P51. So after a few cycles of PCR, let's say maybe 10, I'll transfer all 8 tubes to the P51, turn on the light, and it looks like the fluorescence isn't strong enough to see yet. But that's not a problem, because I can just repeat this process every 2 cycles until eventually I start to see some fluorescence in the last tube. Great! So based on this signal, it seems like there was more RNA for gene X in the last tube than there was in the others. And soon, the other treatment tubes are going to light up too, and that's in sequence according to the amount of RNA for gene X they had in them. And remember, that's the result of cells responding differently to the amount of drug in each tube. So also take note of the 2 controls we used, water and no treatment. They're a baseline to which we can compare the fluorescence because they don't shine as brightly as the rest. And so finally, with just a few snapshots, it's starting to look like my hypothesis is being supported. The presence of medication seems to be associated with increasing expression of gene X in space. So what you've seen here is a really powerful application of a pretty simple tool. And the data gathered about bodily changes in response to medicine may tell us more about what it means to have a safe dosage in space. And so the P51 was a great choice for this experiment, and it could be for yours too, because first of all, it's visually appealing and really easy to understand, and that allows for simple assessments to be made, just like we just saw. Second, it's really, really fast. It takes no time at all to pop the tubes out of the PCR machine, place them into the P51, and turn on the light. You don't even need to wait for the samples to return to Earth to have your answer either. And third, it's versatile. I administer different concentrations of medicine, but you could imagine trying an experiment with treatments over multiple times, or maybe treatments on Earth versus in space, and much, much more. And finally, P51 makes space biology really accessible on the ISS and beyond. All you need is something to take pictures of the results with, like a phone. And so, being that straightforward, who knows? The P51 might just be the DNA detection tool that makes it to Mars. But as the next generation of genes in space participants, that's now in your hands. So good luck and get writing.
Thank you, Christoph. We're so happy you joined us here today to talk about your Genes in Space winning experiment. I hope that Christoph's experiment has inspired you to consider using the P51 in your Genes in Space experiment this contest. To wrap up the webinar, we're going to go through a few questions that you guys have submitted. So the first one is, can my proposal use more than one of the Genes in Space Toolkit techniques? The answer is yes. So Christoph and I both told you about how you can use PCR combined with a P51 fluorescence viewer to estimate how much of your gene or a DNA sequence of interest was present in your sample. You can also combine the BioBits cell-free system with P51 fluorescence viewer. You can use BioBits to produce a fluorescent protein and then visualize it using the P51. Finally, you might imagine how you could combine PCR with a BioBits cell-free system by using PCR to copy a gene and then make it into protein using the BioBits cell-free system. So these are just a few examples of how these tools in the toolkit can be combined. Again, we want to know what's the most creative experiment you can come up with using the Genes in Space toolkit to address your question that you're asking for your Genes in Space proposal. And we think that perhaps you might even imagine how you could combine all 3 tools in the Genes in Space toolkit in your experiment. It's completely up to you and your creativity. The second question is a little bit complicated, so I'm going to read it and then we'll break it down. So the question is, during the finalist launchpad, the finalists proposed both a freeze and fly experiment and an ambitious experiment. Do we need to propose both in our Genes in Space proposal? So as you may know, the 2020 Genes in Space finals had to go virtual this year due to the ongoing pandemic. And they're available to watch on our YouTube channel. I've actually put the link in the description below so that you can go check them out. If you've watched them, you may have noticed that all of the finalists proposed 2 experiments. One is a more simple, what we've called a freeze and fly experiment, and the second is a more complex, ambitious experiment. So the answer to the question is, you do not need to have both of those experiments in your Genes in Space proposal. What we're looking for in your proposal is simply the best experiment that you can design to address the question that you're asking. So most of the time that's a more ambitious experiment. But this brings me to talking through the Genes in Space contest cycle a little bit. So as you guys all know, the contest is currently open. And so now is the time that you develop your idea and submit it to us. You must submit it to us no later than April 12th of this year in order for it to be considered in this year's contest. Once all the applications are submitted, we read through them and we announce awardees, semifinalists, and finalists in the month of May. The 5 finalist teams that we select are then paired with a scientific mentor, and these mentors work with the finalist teams to prepare them for their finalist presentation at the end of July. So the mentor will take the proposal that you submit and help you develop both your simple freeze and fly experiment as well as further refine the experiment you initially proposed. So that's important to keep in mind when you're watching the finalist launchpad, is that all of those finalists had some help from mentors prior to those presentations, and that if you're selected as a finalist, you will have that mentoring as well. After the finalist presentations, a winner is selected, and then that winner works with their mentor and all of us at the Genes in Space team in order to prepare their experiment for space. It will likely launch in 2022. Our next question: does my experiment have to be related to human biology or use human subjects? The answer here again is no. Humans are perhaps the most interesting organism that we're going to send on a long duration space mission, but many organisms will accompany those astronauts as well. For example, we have many microbes in our microbiome that we bring with us everywhere. Additionally, we may bring other microbes or possibly even plants to be used as food or for other purposes. If you're interested in those plants or microbes, you can absolutely use them as the subject of your Genes in Space proposal. You don't have to focus on human biology. But even if you're most interested in a question that relates to human biology, you may not want to use human subjects in your experiment. And the reason for this is that at any given time, there are at most about 6 astronauts on the International Space Station. That's not very many test subjects for your experiment. You may want to rely on model systems. These are more simple organisms that you can use to model a biological process and make a hypothesis about how that process may be occurring in humans. I'm not going to talk too much about model systems today, but I have put a link in the description below to a blog post that one of the former Genes in Space mentors wrote up that details each of the commonly used model systems and talks about how you might use them in a Genes in Space proposal. And so if you're considering using a model system, I really encourage you to check it out. And finally, our last question: do you have any tips on crafting a good hypothesis? I love this question because crafting a good hypothesis is something that we all struggle with. Scientists at many levels have trouble with this. I don't have too many tips on the more formal definition of a hypothesis that you may be familiar with from your science classes. However, for your Genes in Space hypothesis, what we're looking for there is that you have done your research. So what we want to see is that you have read about your topic and understand how this topic, or what is known about this topic here on Earth, as well as anything that's known about that topic in space. And then you've used that research to put together a hypothesis that can be addressed by the experiment that you're proposing. More broadly, if you'd like tips on how to put together a good Genes in Space proposal or just want to tap into some more resources, I encourage you to check out www.genesinspace.org/learn. There's tons of resources there at the page. I'm going to highlight a few of them. The first is our applications tips section. So in our blog, there are many articles talking about what makes a good Genes in Space proposal or various aspects of the Genes in Space proposal. You can access those all at genesinspace.org/learn, and we encourage you to check them out. In addition, we've posted detailed scoring criteria that tell you a little bit more about how each of the questions that you're writing up will be evaluated. So again, encourage you to check that out before submitting your Genes in Space application. Also on our Learn page, you'll find videos explaining various biotechnology techniques, including PCR and DNA sequencing. And you'll also find videos talking about various space biology research topics. So these are interviews with different scientists that are doing biological research on the International Space Station. And we hope that they inspire you to think about different topics that you could use as a source of inspiration for your Genes in Space proposal. And finally, the Meet the Genes in Space Toolkit webinar series. If you haven't checked out the other webinars in this series, we encourage you to watch them, and we hope that they help you as you begin to craft your Genes in Space proposal. I want to wrap up by just reminding you about the details of the 2021 Genes in Space contest. So the contest is open to students attending school in the United States who are in grades 7 through 12. You may apply individually or in teams of up to 4, and your application must be sponsored by an adult. That can be your teacher, your parent, or any science enthusiast in your life. It's free to apply, and your application must be submitted no later than April 12th in order to be considered for the contest. And that's it. That's the end of our webinar. We thank you so much for joining us here today, and we cannot wait to read all of your applications in April. Best of luck on your proposal. If you have any additional questions, we encourage you to email us at genesinspace@minipcr.com. And otherwise, good luck and thanks for joining us.